So students, sorry, sometimes streamings get stopped for today. Please join with the new link and we start again with a fresh start. So, so Bachche, for contacting me, just write me on my email address. Um, Right now, I will be happy to be in touch with you via email. So we are done for today about the assignment and review articles. So let's start uh, with the topic of today. Let's start our workshop day first. So I will be dealing with you basic things here. Uh, so let's continue with the fresh start. Yeah, I trying my best, students, to be louder. So assignment is not compulsory for you. You just have to give it to me via email so that I could see that you are working fine with it. Right, right. Keep the Sunil. Right, you're right. So let's start the workshop for today, or we wait for 2 minutes. At 5:30 we start. So let more people add in. So all these workshops will be on YouTube. So don't worry, you can go through them. So day one, biology, biochemistry techniques, it will be available online. So you can go through them and check it there. So before I start my workshop, uh, assignment for example, okay, sure I can share that also. So assignment example would be like this. Let me check the best one for you. So somebody wrote me each day, like day one, day four, day seven, day eight, day nine, each day. So I cannot share these ones actually because that will be cheating. But I can share you just one day uh, example that how students are making it. cannot find the test, but I will share with you later. It's no worries. MBT internship. Okay, anyhow, I will share with you later about this. So let's continue with the. So review article has to be written by the persons who have done the who are doing the internship. An assignment is not compulsory, but it's for everyone. If you can write, that will be really great for us. So here, before we start, let's check with the cell and size. How cell and size affects the size of each particles. So here you can see coffee bean, grain of rice, time regular point. So you can see the coffee bean to be size of 12 by 8 millimeter, grain of rice to be 8 by 2.5 millimeter, sesame seed to be 3 by 2 millimeter, grain of salt by 0.5 millimeter, amoeba proteus by 500 micrometer, paramecium by your 210 to 60 micrometer. So they're coming in the micrometers, 130 micrometer, the skin cell to be around 30 micrometer, then comes sperms around 6 to 60 by 5 micrometers. So they all are coming in the micrometers, you know. First we start with the millimeters. The chromosome coming out to be 7 micrometer, red blood cells to be 8 micrometer. Further, if we go down, E. coli coming out to be 3 by 0.6 micrometer, lysosomes to be 1 micrometer, mitochondria by 4 by 0.8 micrometer. If we go further, after these bacterium size, shape, morphology, then comes the viruses, which seems to be around nanometers. So measles viruses to be 200 to 20 nanometer, HIV to be 130 nanometer, influenza virus 130 nanometer, hepatitis virus 45 nanometer. Then after nanometer comes the ribosomes around 30 nanometer, antibody bound about 12 nanometer, tRNA about 7 nanometer, hemoglobin about 6.5 nanometer. So if we go further, from nanometers comes your picometers. So adenine, methionine, water molecule, glucose, carbon atom, they all coming out to be in picometers, 340 picometers. So this way, we started uh, with the sesame seeds and coffee seeds and ending at the carbon atom with the picometer. So each basic part, carbons, phospholipids, antibody, all seems to be varied size from your millimeters to micrometer to millimeter. So all things depends upon like this. So this was about the cell size. So based on that, further, let's start with the workshop of our first presentation. Cell is like a factory. So this is an analogy that I have made for you. This is from the childhood, from the 10th class. If you remember how in a cell factory, in a factory, and how our cell, they are both acting like a factory, including the various parts of its. So let's go back in time where we were studying about Golgi body, nucleolus, nucleus, centrioles, microtubules, how they are working together and making the work of cells function good. So how can cells be like a factory? So in a factory, you have many different tasks be completed in the same building. Example, the building supports shelter workers, uh, like workers building goods, maintenance crews, clean up messes, shipping departments gets goods ready to leave the factory and so on. And cell can be thought as a factory with different types, each performing specialized tasks. So cell wall is like a security gate. A security gate protects the factory and helps keep it safe. A cell wall does the same thing for a plant cell. So you see, security gates protect the factory and helps to keep it safe. Cell wall does the same thing for a plant cell. Cell membrane is like front doors. The doors of a factory regulates what enters and leaves the building. A cell membrane does the same thing for a plant and animal cells. So here, this is how the cell membrane works. Then comes the nucleus. Nucleus is a like big boss. The boss controls all the activity inside the cells. The nucleus does the same thing for plant and animal cells. So like it's a big boss where your all DNA material being generated within the nucleus. Then comes the cytoplasm. It's like a factory floor. The floor of the factory has all workers, machine equipment on it. And cytoplasm hold all the organelles in the plants and animal cells. So it's a like cytoplasm large open field. Endoplasmic ER is like an assembly line where the workers do their work. Items move along a conveyor belt to different parts of the plant. ER moves items to different parts of the cells in the same way. So this is the assembly line where all the workers are working in the ER field. Ribosomes are the workers on the assembly line. They are responsible for building the goods in the factory. They are the building proteins molecules. So they are the one who are working on the assembly line. And vacuoles are like the break rooms. In a break room, you have a storage of food items and places to get rid of trash. Vacuoles hold waste, stores water in plant and animal cells. Which cell has one large vacuole? So this is like in a vacuole. And lysosomes are like maintenance janitor crews. In a factory, these guys break down, clean up, and repair items and around the city and the factory. So lysosomes do the same things in the cells. So they are the like janitors who are cleaning and cleaning up, preparing things for your within the cell. Then comes the mitochondria and chloroplast, which are like power stations. The power station supplies energy to the factory, and mitochondria and chloroplast do this for the cells, like a Duracell type. So this is an example of a cell, how a cell is working out in a factory. So this is one example showing you with the restricted area, Golgi apparatus, ribosomes, nucleus, endoplasmic reticulum, mitochondria, membrane bound area. So everything is being adjusted as per the requirements. So one thing you could do if you like, you can go into designing your own factory, make something. You must include all the organelles which we have discussed in that worksheet, and you can choose the job as that what the factory is doing. So something like a ketchup factory, the protein should be the bottles of ketchup is coming out of it. So that was something very short and crisp about the cell before we go along. And so next part, before we take a next part, let's have some video timings. So whenever I'm playing some video, I will be not speaking because I can either speak or video could play. So I will be muting during that time. So I will play what are any. So you all might know aware about DNA and genes. So I will be just sharing you some little bit about these things so that we are all at the same level. So some might be knowing about these basics before already, but some might not be. So main reason of today's workshop is to give you introduction of cells and DNA. So yes, so let's start. Oops, my headphones always get stuck in it. Okay.
The instructions for building all living things, whether a fish, a bacterium, a tree, an insect, or a person, are found in a molecule called DNA. DNA is made up of 4 tiny building blocks. Each building block has a backbone and a chemical base that is represented by the letter A, C, G, or T. DNA molecules have 2 strands that are joined together through complementary base pairing. A pairs with T, and C pairs with G. These building blocks function together in larger units called genes. Each gene is a set of instructions for building a specific protein. A complete set of genes is called a genome. A genome is a set of instructions for building an entire organism. Every person's genome has the same genes arranged in the same order, but small differences in the sequence of the bases in our genes make each person unique.
So that was the basics about DNA, how A, T, C, G, the four subunits, they are making different organisms. We are having different characteristics in each of our cell. So to take that example, let me take you to the same gene, different organisms with this example. Sorry.
These may look like ordinary fish. What you can't see is that they have a gene in them that codes for green fluorescent protein. But shine a blue light on it, and GFP emits a bright green glow. What might surprise you is that the gene for GFP came from a jellyfish. GFP was first discovered in jellyfish over 50 years ago. Since then, scientists have been using it to study all kinds of biological processes. What's amazing is that this gene from jellyfish can be put into just about any living thing. Animals, fungi, plants, bacteria. They can read the DNA code of a gene and build GFP that is identical to the jellyfish protein. That same concept applies to tons of other genes too. The gene that spiders use to make silk webs, for example, can be put into yeast cells. Harvest the silk protein that the yeast cells make, and you've got the raw material to make a strong, lightweight fiber. In fact, a company is using this approach to bring yeast-grown spider silk fabrics to market. How is it then that fish and yeast can read DNA from a completely different organism? It's because, despite their differences, there's something fundamentally the same about how all living things read genes to build proteins.
So here we can see same gene, different organism. So this GFP, green fluorescent protein, could be put in a different organism to check various physiological tests, physiological level of working, and also the spider webs have been put in. They extracted that gene and put them in the yeast cells, and they have created this artificial form of spider webs and created a very nice silk out of it. So this is how you can get the gene and fix into some other organism and can do your things done that as per your likings. So on that note, let's take so it's almost 45. Let's take a 5 minutes break. Just get freshen up. Then we start with the next part of basics of molecular biology. That is your next topic. Okay, then I see you in 5 minutes or so. you, So, students, welcome back to the next part. Uh, mm, Basics of molecular biology. So I will dealing with all DNA and RNA that we have discussed just now, how translation, transcription, how central dogma, how mRNA, ribosomes, how tRNA, they're all doing initiation, elongation, and termination in the process of producing a nice protein, a long polypeptide chain of amino acids. So we will be discussing all these topics here in details. Let's start. So first we start, whenever we are starting with the cell molecular biology, first comes the eukaryotes and prokaryotes. So eukaryotes are the ones which has plant, animals, and fungi, and prokaryotes have bacteria and cyanobacteria. So the cell wall has no cell wall in the animals, but in plant, yes. In eukaryotes, in the prokaryotes, there is a cell wall. Whereas the chromosome segregation, there's a mitotic spindle formation, whereas in prokaryotes, chromosome segregation is cell membrane. Meiosis is happening in eukaryotes, or prokaryotes, it's not happening. Ribosome size is 80S and prokaryotes has 70S. Cell organelles, so nuclear membrane, eukaryotes have positive, whereas prokaryotes is absent in that. Endoplasmic reticulum is present in eukaryotes, in prokaryotes it's absent. So Golgi body, mitochondria, chloroplast, they're all present. So they are nuclear membrane bodies, membrane or body organelles. So they are all present in eukaryotes, whereas the membrane body organelles are absent in the prokaryotes. So what is the definition? Molecular biology is the study of molecular underpinning of the process of replication, transcription, and translation of the genetic material. So all your replication, transcription, and translation that is happening to create a genetic material is known as molecular biology. And they are it's shown that how plants, microorganisms, molecules, population, animal cells, they're, they're all interlinked with each other like this. This field overlaps with other areas of biology and chemistry, particularly genetics and biochemistry. Molecular biology chiefly concerns itself with the understanding of the interaction between the various systems of cells, including the interaction between DNA, RNA, and protein biosynthesis, as well as learning how these interactions are regulated. Much of the work in molecular biology is quantitative, and recently much work has been done at the interface of molecular biology and computer science and bioinformatics and computational biology. Since the late 1950s and 60s, molecular biologists have learned characterized to isolate these molecules like DNA, RNA, proteins, and major other structural enzymatic molecules of animal cells. So these are the major components involved in the molecular biology, that is DNA, RNA, and protein. So gene, which is a unit of hereditary units. So DNA segments that carries genetic material are called genes. It is normally a stretch of DNA that codes for a type of protein or for a RNA chain that has function of the organisms. Genes hold the information to build or maintain an organism cells and pass genetic traits to offsprings. So deoxyribonucleic acids, DNA is a nucleic acid uh, that contains instructions used in development and functioning of the all-knowing living organism. So DNA आपका बच्चों is the main nucleic acid that contains all the genetic instructions and functioning of the all living organism and some viruses also. So it is a blueprint needed to be construct other than components of cells such as proteins and RNA of molecules. So that is DNA. So there are two strands makes the shape of double helix. There are two strands. We have watched that in the video. Two strands run in opposite direction to each other and therefore anti-parallel. They run in opposite direction of each other, so they are anti-parallel. Chemically, DNA consists of two long polymers of simple units called nucleotides. So these are nucleotides with a backbone of base of sugar and phosphate. So you have a sugar and phosphate group, and these are nucleotides being attached. So like this. So your nucleotides is a sugar, phosphate, your nucleoside bases, nitrogenous bases pairs. So all these three together makes your DNA double helix, or you can say nucleotides. Nucleoside is sugar plus base, that is nucleoside, whereas phosphate plus sugar plus base is equal to nucleotide. Phosphate plus sugar plus base is equal to the nucleotide. So what are the bases that we are using here are types of adenine guanine, that is fused 5-6 member heterocyclic compounds, that is purines. So these are your adenine and guanine. There is also then cytosine, thymine, and then this is uracil. So they are the 6 membered rings of your pyrimidine. So purines are 5 or 6 membered heterocyclics, and cytosine, thymines are 6 membered rings. The fifth pyrimidine called uracil, which is happening in the RNA only, differs from the thymine group by the lack of methyl group on its ring. So A pairs with T, G pairs with C with a double bond and triple bond respectively, but whereas A pairs with double bond with the U. So DNA double helix is stabilized by hydrogen bonds between the bases attached to the two strands. So one major difference between DNA and RNA is the sugar. So with the two deoxyribose sugars in DNA being replaced by the alternative pentose sugar ribose in the RNA. So here you can see deoxyribose sugar, where you have at the second carbon there is no oxygen present. So this is a deoxyribose. But if it is oxygen is present like this, it will be a ribose sugar, which is present in the RNA, and that's the major difference between DNA and RNA. And this is how showing adenine and thymine, they are making double bond, and in the guanine and cytosine they are making a triple bond together. So and these bonds are hydrogen bonds actually. Further size of it, um, so between two base pairs, between the length is 0.34 nanometer, the diameter is around 2 nanometer. There are major group, minor group. These are base pairs, and this is on the side is a sugar phosphate backbone. So DNA chain is around 22 to 26 angstrom wide, and one nuclear unit is 3.3 angstrom long. So DNA is biologically important type of molecules that consist of long chain of nucleotide units. Each nucleotide unit consists of nitrogenous base, ribose sugar, and a phosphate. So double-stranded RNA. Double-stranded RNA is RNA with two complementary strands similar to the DNA found in all cells. Double-stranded RNA forms the genetic material of some viruses, that is double-stranded RNA viruses. So So there are types of RNA, that is messenger RNA, ribosomal RNA, transfer RNA, that is abbreviated as mRNA, rRNA, and tRNA respectively. And their respective function is to have codes for protein, translation, and translation further. And they are present in all organisms. So in the post-translation modifications, there are smaller nuclear RNA, that is yRNA, telomerase RNA, that is RNA they are called as. The sRNA has function of splicing and other functions, eukaryotic and archaea. YRNA has a function of RNA processing, DNA replications, and they are present in the animals only. And telomerase RNA is used for telomere synthesis, most eukaryotic working. So regulatory RNA, antisense RNA, aRNA, transcriptional attenuation, mRNA degradation, mRNA stabilization, translation block. So they're all being functioned in the all organisms in this case. So messenger RNA mainly carries information about protein sequence to the ribosomes. Before I continue, let me take you the story of transcription and translation, because that will make sense, that will make the story join together and make things more better. So let's play a game first, a little bit game. Who likes games? Everyone likes games. So let's build a DNA. So here we have to drag T in front of T, it would be A. So we are building a DNA. And in front of G is C. In front of C is G. In front of A is T. G is C. T is A. C is G. T is A. C is G. T is A. A is T. Like this, we continue. We keep continuing. C is G. So by this, if I keep continuing, it will take me 50 years to transcribe this DNA into one human cells. So it will take 50 years to do so. So hope you can understand that. So if I keep continuing, T is A, and like this it will keep continuing. Just remember, in the case of A, it will be U in the case of mRNA. So what's so a small video about protein.
Proteins carry out most of the body's functions, and they make up the bulk of its structure, from hair and fingernails to bone and muscle. In fact, nearly everything in our bodies is made from or by proteins. The instructions for building proteins are found in segments of DNA called genes. Proteins are made of long chains of building blocks called amino acids. There are 20 amino acids in all. Put together in different combinations, amino acids form proteins with a broad variety of shapes. This diversity of form gives proteins their many functions. The proteins a cell makes determine what it can do. Our cells are constantly at work, producing the thousands of different proteins that keep our bodies functioning.
So small video about how proteins are functioning in our body in a very elegant way. So it was a nice one to just to have an introduction about proteins. So now let's start the other part, transcription and translation.
all living things use. Transcription and translation are the processes that all living things use to read the information in DNA and build proteins. Each gene in a cell's DNA codes for a specific protein. During transcription, cells make a copy of a gene out of mRNA. During translation, they use that mRNA copy as a blueprint to build proteins out of amino acids. These proteins perform the tasks that make living things function. The flow of genetic information from DNA to mRNA to protein is central to life.
So,
Inside the nucleus, RNA polymerase attaches to a gene. It separates the two complementary DNA strands, exposing the gene's unique sequence of bases. When the end of the gene is reached, transcription is complete. The new strand of mRNA passes out of the nucleus and into the cytoplasm, where translation takes place. The instructions in an mRNA strand are read in groups of 3 bases called codons. Each codon must be translated, one after another, in order to build a functioning new protein. The key structures in that process are the ribosome and transfer RNA, tRNA. Each tRNA molecule has its own anticodon and a matching amino acid, the building blocks of proteins. To begin translation, a tRNA molecule coded for the amino acid methionine docks with a ribosome. The ribosome attaches to the end of the mRNA strand. It slides along until it finds a start codon, AUG. Through complementary base pairing, the start codon pairs with the anticodon on the tRNA molecule, signaling the beginning of the protein instructions. The methionine molecule is now the first amino acid in the new protein. The ribosome shifts to read the next codon. Each codon is recognized by one of 20 different tRNA molecules. Each type of tRNA carries a different amino acid. The ribosome continues moving along the mRNA strand, one codon at a time, matching it to a complementary tRNA. It adds each amino acid to the growing protein in just the right order. The sequence of amino acids determines the final shape of the protein and the function that it will carry out. Give translation a try. When the ribosome reaches a stop codon, it detaches from the mRNA strand. The chain of amino acids folds into its final form and the new protein is complete. This protein, DNA helicase, will now slide between two complementary strands of DNA, temporarily separating them so that they can be copied. The incredible processes of transcription and translation enable cells to carry out all of the functions necessary for life.
So students, that was about a very short and very nice trip about transcription and translation where your all concept might have been clear. Now before I forgot these things, let me share all of them on Telegram. This one, right? October 20. Okay, so we have seen that how messenger RNA helps in producing your taking this messenger RNA and going to the nucleus from outside the nucleus and then starting to working for transcription. So mRNA is then exported from nucleus to cytoplasm where it is bonded to the ribosomes and translated into corresponding protein from with the help of tRNA. In prokaryotic cells which do not have nucleus and cytoplasm compartments, mRNA can bind to ribosomes where it is being transcribed from DNA. So this is your transfer RNA. It is a small RNA chain of about 80 nucleotides that transfer specific amino acids to a growing polypeptide chain at the ribosomal site of protein synthesis during translation. So this is how it is shown here, the codons of the transfer RNA where it is binding it. Then this is a clover leaf shape of your tRNA where they're making ester bond with the amino acids on the top and an mRNA is being attached at the bottom. And they are attached here also by the intermolecular base pairing. So it has a site for amino acid attachments and anticodons region for the codon recognition, and that site binds to specific sequence of the messenger RNA through the hydrogen bonding. So ribosomal RNA is the catalytic components of ribosomes. So eukaryotic ribosomes contains 4 different ribosomal RNA molecules, that is 18S, 5'S, 28S, 5S RNA, and they are synthesized in the nucleolus. So you can see here like how ribosomal RNA of 70S and 80S is being made. And in the cytoplasm, ribosomal RNA protein combines to form nucleoproteins called ribosomes. The ribosomes bind messenger RNA and carries out protein synthesis. Several ribosomes may attach to the single mRNA at any time. So ribosomal RNA is extremely abundant and makes about 80% of the 10 milligram per ml of RNA found in the typical eukaryotic cytoplasm. So there is a difference between DNA and RNA. So RNA nucleotides contains ribose sugar, uh, and DNA contains deoxyribose sugar. RNA has a base uracil. DNA has a base thymine. Presence of hydroxyl group at the second position of the ribose sugar, and DNA is a lack of hydroxyl group at the second position of the ribose sugar. RNA is usually single stranded, where DNA is your double stranded. So that was something about DNA and RNA. We have discussed about messenger RNA, tRNA, how